Good afternoon. I'm Dr. Brian Shapiro, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Physiological Relevance or Ease of Use. Enjoy both with hTERT-immortalized primary cells presented by Mr. Kevin Grady and Dr. Chaozhong Zou. Mr. Grady is the Senior Product Line Business Manager at ATCC Cell Systems, while Dr. Zou is a Senior Scientist. In today's webinar, Mr. Grady will discuss our broad portfolio of hTERT-immortalized primary cells and provide application data to illustrate how these cell models can easily be incorporated into your workflow. Dr. Zou will then present data showing how ATCC's new kidney transporter models can be used for predictive toxicology testing. If you have any questions for either speaker, please use the chat function available through the webinar program. Questions will be answered at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. With that, I would like to welcome Mr. Grady.
Thank you, Brian. So before we get into the presentation, I want to give a little bit of background about ATCC. ATCC was founded in 1925 as a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and services center in Gaithersburg, Maryland. ATCC has achieved worldwide brand name recognition for both its quality and diversity of materials that it supplies. We're the world's premier biological materials resource standard development organization with over 4,000 cell lines, 70,000 microbes. ATCC collaborates with and supports the scientific community with industry standard and innovative biological solutions. We have a growing portfolio of products and services with sales and distribution in over 140 countries, including 12 international distributors. ATCC has a team of over 475 employees, with over 1/3 having advanced degrees. ATCC has achieved multiple accreditations, including ISO 9001, as well as ISO 13485. So as Brian talked about earlier, the agenda we want to focus on today is we're going to go through ATCC's hTERT-immortalized primary cell portfolio, and then I'll take care of that portion of the presentation. And then I'll be turning the presentation over to Dr. Zou, who will go through our recently launched kidney transporter models and provide a background on those cells, as well as some application data that we've generated in-house, as well as some that's been generated from outside collaborators. So everyone's familiar with ATCC for continuous cell lines, such as HeLa cells, JERCAD, and HEKs. But today I want to focus on ATCC being a complete solution supplier for your research needs for cell biology. As your research develops, you'll likely need models that are have more biological relevancy. ATCC can supply these more biologically relevant cells in the form of either primary cells or hTERT-immortalized primary cells. Both of these cell types are held to the same high-quality standards associated with ATCC's continuous cell lines, such as reliability, authentication, and performance. Okay, so as a researcher, you're faced with the question of which model is the best for my research needs, as each of the different models that I'm going to go through has both positive and negative features. Starting with cancer cells, which are the cell lines that are most readily associated with ATCC. The positive features are they have high proliferative capacity, they're generally easy to culture, and because of their clonal nature and proliferation, you can generate lots of cells without introducing experimental variability. However, all those positive features are based upon the fact that these are isolated from tumors, so they have low biological relevancy to normal tissue or normal physiology. Next are cell lines that are generated from cells being transfected virally with oncogenes. The resulting cell lines do have a good proliferative capacity, but due to the presence of one or more oncogenes, the cells still exhibit low genetic stability, which leads to poor predictability and low biological relevance. Next, moving up the biological relevance value chain is hTERT-immortalized primary cells. These cells exhibit normal genetic stability, with near primary cell performance while exhibiting the high proliferative capacity of a cell line. So you can culture a large number of cells for your research with no concerns about introducing experimental reproducibility, thus providing a great model for tox studies or any large screen that you may need to run. The last model to discuss are the primary cells. These are isolated directly from normal tissue. As such, they exhibit the highest biological relevancy, which best mimic an in vivo response. The major drawback with using only primary cells in your research is you can only obtain a certain number of cells from a specific tissue, and these cells have a finite lifespan, and therefore cannot be expanded to any great cell numbers. So if your research requires a large number of cells, you will be using cells from multiple donors and introducing biological variability. So as we're going to focus on hTERT-immortalized primary cells as an optimal model for cell research with high biological relevancy. To make an hTERT-immortalized cell, you start with a primary cell and stably transfect in the telomerase gene. In the graphic on the right, we see what happens as a normal somatic cell grows. As it grows and divides, the telomere decreases until the telomere sequence is reduced to a point where the cells enter senescence. In the lower graphic depicts what is happening in cancer cell growth. One aspect of cancer is that the cells divide and the telomerase continually rebuilds the telomere and the cell never enters senescence. So hTERT-immortalization makes use of this genetic modification to the telomerase enzyme. Primary cells are immortalized by transfecting the sequence for telomerase into the cells and selecting for the cells that stably express telomerase. The hTERT-immortalized cells provide you with a model that offers the growth characteristics of a continuous cell line with the performance of a primary cell. ATCC stringently validates the performance of our hTERT-immortalized cell models, confirming the performance via surface marker expression, karyotyping, and biological performance. These characteristics are also confirmed at longevity, which is in excess of 35 population doublings past the creation of the master cell bank. In fact, some of our hTERT-immortalized cell lines have been tested at an excess of 50 passages with no difference in performance noted. Now I'll briefly introduce a few of our cell models from our portfolio of hTERT-immortalized primary cell lines. First, I'll introduce our skin models. We have a full complement of cell types from fibroblasts to keratinocytes, melanocytes, and dermal endothelial cells. These can be readily co-cultured to create a complete skin model in a dish. In this slide, this data actually illustrates such a co-culture experiment. In this case, we added dermal fibroblasts as well as keratinocytes. In the DAPI stain micrograph, you can clearly see the two distinct layers of fibroblasts on the bottom and keratinocytes on the top. To achieve this model, keratinocytes were cultured on a collagen raft embedded with dermal fibroblasts at the air-liquid interface for 11 days post-airlift. At that point, we wanted to look for various markers for the keratinocytes. So in the micrograph here, we're staining with an antibody against KRT14, which is a well-established marker for keratinocytes. You can clearly see that throughout the keratinocyte layer, looking over here, that this layer is clearly expressing the marker for keratinocyte. And there's a high level of expression throughout the entire layer. Filigran, in this micrograph down here, is a marker for terminally differentiated keratinocytes. And as the overlay clearly shows, that the outermost section of the skin model consists mostly of fully differentiated keratinocytes expressing the filigran. Thus, we've created a fully differentiated skin model in a dish. Next, I'll introduce our airway models. We have a full complement of primary cells, such as different epithelial cells from different regions of the cells, smooth muscle cells, lung fibroblasts. Regarding hTERT-immortalized primary cells, we have lung fibroblasts, bronchial tracheal epithelial cells, and small airway epithelial cells. The next model I'd like to introduce is our urogenital models. We have a broad and diverse offering for primary cells, as can be listed here. But regarding hTERT cells, we focused on two main tissues: cells isolated from the endometrium and three different cell types isolated from the prostate. Our prostate lines offer a unique opportunity to study the tumor microenvironment. So our 3 lines from the prostate are normal associated fibroblasts from normal prostate, cancer associated fibroblasts from a donor suffering from prostate cancer, and then normal prostate epithelial cells. So in the co-culturing model, we took the normal prostate cells, normal prostate epithelial cells, and cultured them with normal associated fibroblasts or cancer associated fibroblasts, and then looked at the growth proliferation. And as you can clearly see down here in the stain, the epithelial cells that were grown in co-culture with the calcium associated fibroblasts exhibited much greater proliferation, as noted by the many more cells that are seen there. So now we wanted to check, we've shown that this co-culture model and microtumor environment can be studied with primary cells. We wanted to look at what happens with some of our established cancer cell lines. So we wanted to see if the microenvironment had similar effect on some common cancer cell lines, as is illustrated over here on the right. We obtained mixed results as some of the cell lines exhibited proliferation when co-cultured with cancer-associated fibroblasts, that would be the green bars, and those in co-culture with the normal associated fibroblasts, the blue bars. So when co-cultured with cancer-associated fibroblasts, lines such as DU145, which is a prostate carcinoma, and A549, which is a lung carcinoma cell line, did show increased proliferation in the presence of our cancer-associated fibroblasts. This cell line, RWPE1, which is a prostate line that was created through HPV transfection, did not exhibit similar cases. So the question of tumor microenvironment effect is currently being further investigated by ATCC R&D. Mesenchymal stem cells are widely used in cell biology research. ATCC supplies primary adipocytes as well as 3 forms of primary MSCs, isolated from adipose, bone marrow, and umbilical cord. As for hTERT-immortalized cells, we currently have 1 MSC line available, a line created from adipose-derived MSCs. For all of our MSCs, we test for surface marker expression for the full spectrum of markers as provided by the International Society for Cellular Therapy. As you can see in the data table supplied here, all of our cells meet the ISCT guidelines, primary cells as well as the hTERT-immortalized cells. As I stated earlier, our hTERT cells can be used in co-culture to create more in vivo-like models. ATCC R&D have developed a system for studying angiogenesis. They combined an endothelial cell with our hTERT MSC. The system is known as our Angio-Ready System. Phase contrast micrograph here shows angiogenesis occurring with the endothelial cell network developing on top and among the supporting MSCs. The GFP micrograph over here shows the developing endothelial network. Since the hTERT endothelial cells used in this system stably express GFP, analysis can be done live and in real time. As we worked with this co-culture model, we noticed that lining up along the developing endothelial tubules were cells that expressed smooth muscle actin, a marker for smooth muscle cells. We did not add smooth muscle cells to the mix, so the MSCs in the proximity of the endothelial cells are either developing into cells or causing cells to develop into cells that express smooth muscle actin, giving you a near in vivo model for blood vessel development with an inner layer of endothelial cells surrounded by smooth muscle cells in a stromal matrix. To verify that this model has a high level of predictability, we added increasing doses of a known anti-angiogenesis drug, and we observed a dose-dependent decrease in tubular formation. So the drug we added, Sunitinib, which is a known anti-angiogenesis drug, and you can see that as the doses of the drug increase, the amount of tubular formation increases, as does the amount of cells that are expressing smooth muscle actin. So this is a very good predictive model for the study of angiogenesis. Lastly, I'd like to briefly introduce our ATCC kidney models. We offer several types of primary and renal epithelial cells and hTERT-immortalized renal proximal tubule cells. Primary kidney epithelial cells in culture quickly lose the expression of key renal transporters. So ATCC recently introduced a portfolio of hTERT cells that overexpress the transporter proteins OAT1, OCT2, and OAT3, thus creating a model that would be usable for critical studies of toxicology as well as kidney physiology research. And with that, I will now turn the presentation over to my colleague, Dr. Zou.
Thank you, Kevin. Good afternoon, everyone, and thank you for taking time attending this presentation. Now I'm going to introduce you the ATCC just launched next generation in vitro renal transport cell models. Here is the agenda. So we will go a review of current in vitro transport cell models. And then I will tell you the story of how we generate the RPTEC renal transport models and how we can use those transport models. So first, we all know that kidney is one of the major target organs for the drug-induced toxicity because the kidney has a large functional reserve and nephron toxicity effects sometimes become obvious only after regulatory approval. Those nephron toxicity potential often underestimated when new drugs are available. One example would be the COX-2 inhibitors, which leads to clinical complications. So the reason for why the kidney has this function is because the renal proximal tubular. So show in the left, this blue rectangle area, like the proximal convertible tubes. Here is a major target for drug-induced toxicity because it's a filtrate concentration and it transport our drugs and organic compounds. So we just mentioned that in the proximal tubular areas, there is a lot of the transporters can transport the organic proteins and other small compounds. So let's look at all the transport in the kidney. So in the right side of the drawings, there is a lot. In the kidney there is two kinds of major transport. One is called the ATP binding cassette transport, or also termed as ABC transporters. Another one is solute carrier transporters. So I use a red rectangle to circle some of the very important SLC transporters. So today my talk will only focus on the SLC transporters. And a lot of regulatory documents published by FDA, EMA, and ITC recommends evaluate OAT1, OAT2, and OAT3 as the major SLC targets for drug interaction testing expressed in the kidney. So my talk will also focus on OAT1, OAT2, and OAT3, these three major SLC transporters. So we all know that cell-based models are still one of the best valuable tools during the drug discovery stage and very useful for preclinical testing because it's easy to use and can do high throughput screening. In the next several slides, I'm going to take you to a short journey to search for the best in vitro kidney cell-based models. So for a good cell-based models, we are looking for the high predictability. So which means the cell has a matured kidney proximal tubular origin and can best mimic the proximal tubular microenvironment. Let's start with the primary cells. As Kevin just mentioned, the primary cells can make excellent models, in vitro models, because they often best represent the in vivo tissue. However, in the case of kidney cells, primary RPTEC cells quickly lose the expression of the OAT1, OAT2, and OAT3. These are three most important SLC transporters in culture very quickly. Therefore, they are not relevant models in this particular case. Moving next to continuous renal cell lines. For many years, there have been a number of renal lines available from a variety of species. However, you can see from this table, even after all this time, none of these has been fully characterized to show that they recapitulate the major characteristic of the proxitubular cells. The next step in the evolution of renal models is to use gene modification to overexpress the transporter genes in either non-kidney or embryonic kidney cells, such as HUK293 cells. So ATCC had a first generation of these models by overexpressing the OAT1 into the HUK293 cells. However, those first generation models still lack the critical proxitubular microenvironment characteristic. To address this and develop more physiologically relevant next generation renal models, my lab started with an immortalized proxitubular cell line, then stably integrated SLC transporters into these cells. This will be the next generation cell models. I will now walk you through the generation and the application of those lines. So let's first spend a little bit of time introduce you the cell line we are using. So it's called immortalized human adult proximal tubular cells. We termed as RPTEC TERTI. This cell, the primary RPTEC cell, was derived from the normal adult renal proxitubular epithelial and immortalized using hTERT only. And you have been hearing from Kevin, hTERT by far is the most physiological relevant method to immortalize the cells. And we fully characterize this cell line. You can see from the right some of the pictures or figures showing that it retains its uniform expression of ECAD hearing and the CD30, the red color, and most importantly is form domes in culture. So what is domes? Domes are multicellular cystic structures which have been described previously in epithelial cultures from various tissues that have a non-transport or secretory function in vivo and in vitro. So that means this hTERT immortalized lines retains the most important, the PT epithelial function. And also it has stabilized tier called trans-epithelial electrical resistant, the characteristic. So we have this line. So next, but unfortunately, this line, as well as its primary cells, lost the OAT1, OAT2, and OAT3, these most important SLC transporters expression in culture very, very quickly. So our strategy is by overexpress those important transport back into this valuable cell line. So we transfect OAT1, OAT2, the human OAT1, OAT2, and OAT3 genes into the TERT1 cells and generate a stabilized individual clones for OAT1, OAT2, and OAT3. So after generate these cell clones, the first thing we need to verify how those genes are expressed. Let's look at the OAT1. So we use 3 different methods to verify the OAT1 expression. In figure A is we choose four different clones and doing the RT-PCR to verify the mRNA expression of AUTO1. And you can notice the parental line doesn't have has unnoticeable expression of the AUTO1. And we are in the figure B, we use the Western blot to verify the protein expression. Also, again, the parental line doesn't have any or unnoticeable AUTO1 expression. And importantly, we did the immunostaining to verify the overexpressed AUTO1 is correctly localized to the cell membrane. And we also, in a more step, showing that did the sequencing of this overexpressed proteins shows no mutation, and also by ddPCR verified that there is 7 copies of AUTO1 into the cells versus the 2 in the parental line. Secondly, we use the same method to verify the OC2 expression by figure A for using the RT-PCR to verify the mRNA expression. The figure B, is use the Western blot to verify the protein expression. The figure C, the upper panel is the green color, is a localization of the overexpressed OC2, and compared to the parental line has a unnoticeable the OC2 expression. Now come to the OAT3. We use 2 methods to verify the OAT3 expression. One is by RT-PCR to verify the MR expression, and second is by immunostaining to verify the overexpressed OAT3 is expressed, localized to the cell membrane. By comparing to the parental line, you can see unnoticeable OAT3 expression. So we verified the expression of those 3 genes by molecular method as well as biochemistry as well as the immunostaining. So next we will see if those clones still retains the important proxitubular epithelial cell characteristic. The first thing, as we have been shown before, we check the dome formation. You can see from these 4 pictures, the first is parental, the second one is OAT1 and OAT2 and OAT3, those clones we have been choosing. They formed the domes very efficiently as comparing to the parental line. So again, the domes are very important feature of non-transport and secretory epithelial the characteristic. We also move in a more step to stain the key markers of the parental line, such as CD13 and ECAT hearing. So the first panel is the OAT1, second one is the OAT2. By comparing to the parental line, they show similar these 2 key markers staining. This page shows the OAT3 staining of CD13 as well as ECAT hearing. By comparing it with the parental line, those shows very similar these key markers staining too. So the above several slides, we verified the expression as well as the localization and the key, and showing that those clonal cells retains the major important characteristic of the parental markers. So now we want to ask, are those expressed protein function as it should be? So because those are transporters, so we want to verify it has uptake. It can transport specific substrate. So before we go, so I want to introduce a little bit of our protocol. So we use 2 kinds of assays. One is uptake assay, another inhibition assay to validate those overexpressed cells has the uptake function and specific. So we first see the equal number of the cells in the 296 well plate in triplicate and incubate for 24 hours. Then for the uptake assay, we do the increasing concentration of the substrate, either 6 CF or EM1, and incubate for 20 minutes at 37 degrees. Then we washed with cold HBSS 4 times. Then we lyse the cell and the fluorescence intensity was measured. For the inhibition assay, we seed the cell similarly as the uptake assay, and during the inhibition assays, we using increasing concentration of our inhibitors with the constant concentrations of uptake substrate and incubate for 20 minutes. After 4 times of the cold HBSS wash, cells will lyse and the fluorescence intensity were measured. So let's look at the data. So the first thing we want to verify all O21, O2, and O3 all has very high has the uptake sensitivity. So what we do is using the fluorescence tagged small molecules, which is known specific are the substrate for O21 and O3. Such as the 6CF and the EAM1 for O2. You can see from the bottom, this 3 table, even at very low level, begin from a very low level of the substrate, the uptake ratio is already very high, and from above all of these, the figures you can see with the increasing concentration of the substrate, the uptake activity is very high. So which means those all 3 of those cells display very high uptake activity. So next question we want to ask is, are those activity specific? So what we so in order to answer this question, so we use the substrate specific, the transport specific inhibitors to inhibit the uptake. So we use probimicid and lovobicin for O21, and sunitinib and lovobicin for O3, and then cimetidine and quantidine for O2. You can see from all of the figures displays like with the increasing amount of all the inhibitors, the uptake activity is decreasing, and all the IC50 is in line with other published data. So these 2 slides shows the uptake cells we developed has a high uptake sensitivity and the uptake is very specificity. Those are by using the fluorescence tagged substrate. The reason was that this the fluorescent tag is easy to use, and most of the lab can perform that without additional instruments. We also have other method to verify our the cells, the uptake activity. Here I'm showing you our collaborator in the Ohio State University. They did the uptake as well as the inhibition assay for the RPTEC OAK2 by using 2 substrates. One is TEA and another is beta-formine. They labeled this, the radio labels both of the substrate. And these 2 graphic, you can see the parental line is dark, and the OAK2, the uptake activity are shown as a gray. And then they use they use 3 different inhibitors. The first one is known ABC transport inhibitors, with here as a non-specific inhibition, and the second one and the third one are known to inhibit the OAK2 activity. So you can see from both of the figures. So at the DMSO and level, you can see the OAK2 has a very high uptake activity compared to its parental activity. Here is what is shown here, as well as in metformin, the uptake activity, and the non-specific inhibitors couldn't inhibit the OAK2 uptake of either TEA and or metformin. But the other inhibitor who is specific to the OAK2 shows a very good inhibition activity for both of the substrate. So this confirmed in the more step that the OAK2, the models we generate should be has its very good function as it's supposed to be. So next, I will show you how our models, show you one example of how our cells, the model kidney model, can be used for nephron toxicity studies, such as you screening a lot of compounds, those kind of things. So these the following data is produced by our collaborator in Merck, the farm. They used this to screen their compounds. So here is a brief protocol for cell viability assay. They seeded around 35,000 cells in tripleted 96 well plate, incubated overnight. Then they incubated with a series of compounds at various concentrations, as indicated for 3 days. The cell viability was measured using the Prometheus cell titer globe. This is the data showing the one drug telephone, which is known is toxic and is a substrate of O2-1. You can see with the increasing concentration of this drug, the drug is toxic, cause more than 40% of the O2-1 cell death, but barely can cause the RPTEC, the parental line cell death, which means the cells uptake this drug and this drug causes the cell death. And interestingly, if we blocking the MRP4, which is the efflux transporter by another drug called Cifrin 2, this one will further decrease cell viability. The explanation could be it's blocking it's increase the telephone the concentration in the cell that even cause more cell death. Secondly, they used another drug, acetylform, which is also known. It's cause cell death via the O2-1 pathway. So you can see from here with the increasing amount of the drug, it caused more than 50% of the RPTEC cells death, but it caused very little or barely can cause the RPTEC parental line, the cell death. This is confirmed in the another step that the O2-1, the cell models, is very specific and can be used for the nephron toxicity study. So from these two studies, we concluded that the Merck, the RPTEC TERT-1 O2-1 cells, but not the parental RPTECs, demonstrate decreased cell viability following treatment of telephone, a substrate of O2-1 and O2-3. If we blocking the MRP4, the efflux transporter of telephone by another drug called Cifrin 2, further decreases cell viability, presumably through increasing the drug concentration in the cell. So, and all three, the compounds are toxic to the RPTEC O2-1 cells. So this is in a more step confirming the O2-1, at least showing the O2-1 example can be used as a very good model to screen your compound, which is could cause the nephron toxicity. So here is one example we propose. You can use the RPTEC renal transport models for your either study or in the screening during your drug discovery stage or preclinical toxicology the stage. So you add your candidate compound screening against either OAT1, OAT2, or OAT3, and then determine is the uptake inhibition assay or the viability assay to determine if no change in the transport or the viability. Further testing is needed to determine the mechanism. If positive or negative change in the transport, then you will be able to determine your candidate compound is through which pathway or is toxic to the kidney. So here is this page is a summary. The RPTEC TERT1 renal uptake cell models stably overexpress OAT1, OAT2, and OAT3. The expression has been confirmed by PCR, Western blot, ICC, and we also verified the copy numbers by digital PCR. And importantly, the clonal stable cells retain important characteristic of in vivo adult renal cells. The performance of these stable cells are well characterized by 6-CFEM1 uptake and inhibition assays, also confirmed by radio-labeled TEA metformin and PAH uptake assays. And we also tested against the well-known renal toxic drugs, showing that those are good models for renal toxicity screening. And also, in addition to all of the hTERT-immortalized cells we have been talking about, we also have resources available for you to use or download, such as the Immortalized Cell Culture Guide or hTERT-immortalized Cell Brochure. And for more information, you can visit the ATCC website. Here, showing the two examples of the cell culture guide and the primary cells catalog. Given that, so I will hand the presentation to Brian.
All right, well, thank you, Dr. Zou. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. This session will be documented as a web page and archived along with the recorded webinar presentation on the ATCC website at www.atcc.org.